Okay, let's talk about different types of neurons and then how neurons communicate, send messages within a single neuron and between different neurons. So, there are many different types of neurons, and I'm not even getting here in this unit into the different kind of like shapes and subtypes of neurons. There are many that look just like I have shown you here with a nucleus and dendrites on one end and an axon on the other. But there are other types of neurons that, for example, don't have myelin sheaths or have a cell body that's kind of in the middle somewhere. They're all different kinds of shapes, but this is kind of the prototypical sort of neuron. And when we get to the different, I guess, classes or different functions of neurons, we'll talk about sensory neurons, interneurons, and motor neurons. So sensory neurons are the ones mostly at the periphery of your body, like your fingertips or other parts of your skin, that are basically getting sensory input from the environment. So the neurons in your eyes, again, or your fingertips or something like that. So they're getting, pressure or temperature or light information, and those messages are then going toward the spinal cord and brain, so you can come up with some sort of reaction to those things. Now, sensory neurons don't always send those signals right to the brain. For example, if the reflex of if a doctor taps on your knee with that little hammer thing that I can't remember the name of, the reflex of your leg shooting out is not something that involves your brain. That just is a direct connection between your sensory neurons that input and the motor neurons that are dealing with that movement. So the brain doesn't always become involved. We do have some kind of automatic sorts of behaviors. Interneurons are the ones that are the most plentiful. So you have these sensory neurons on the periphery of your body, and you have interneurons taking up most of the space in the pathways, carrying information between different processing sites and your brain, spinal cord, and all these other neurons. Finally, we have motor neurons, and those are the ones that are carrying commands away from your brain toward, different parts of your body. So your motor neurons taking the message to move your leg or grab something with your hand and extend or contract those types of muscles. So sensory neurons on the periphery getting information, motor neurons sending that information out, and interneurons taking the messages in between all the different sites. Now, there are many other different names for these things. For example, sensory neurons are also called afferent neurons. Motor neurons are called efferent neurons. So the difference between an A and an E there, I think that makes it really confusing. So I'm going to stick with sensory and motor instead. But, there are different types of terminology for these different things. Okay, so I've shown you this basic picture before, but I want to show it again just to show you, again, the direction that the neurons are basically communicating. And again, this is just one basic type of neuron. There are other ways this might happen, but mostly the neurons are getting information from from other neurons at their dendrites. They are then kind of summing up that information from all these different dendrites, and they might then send a message down the axon to the axon terminals to send messages to neurons further down in the process. So this is the direction of communication generally that's happening within a neuron. But how does it send this information then? I think it's very intuitive for people to think about it like an electrical impulse. And that's not exactly correct. It's not a purely electrical impulse like, you send a charge down a wire. This is something a bit different, an electrochemical impulse. It's still dealing with charges, but those charges, rather than just being through a solid state like a copper wire or something, they're being sent by changes in chemical composition and balances on the inside and outside of that axon. So again, this electrochemical message, it's not just electricity, it's flowing chemicals in and outside of your axon. So it's easy to think about, or it's easier to think about this in terms of a change in charge for your neurons. So your neurons, when they are not active, basically, when they are resting, they have something we call a resting potential. And this means that the polarization or the charge of your axon, the inside of your neuron, is about 70 millivolts, very small negative charge, negative 70, relative to the surrounding area. So the chemicals outside of the axon are charged more positively than the chemicals on the inside by about 70 millivolts. So on the uh, outside of the neuron, then you have more positively charged chemicals, and on the inside, you have more negatively charged chemicals. And these are different things like sodium, potassium, and chloride mainly. There are other things around, different parts of your body, but these are the primary positively and negatively charged ions that are the molecules that are spreading around through there. So these naturally are going to try to even out and reach a sort of neutral charge. But your axons have these different pumps or ion channels that are going to allow different ions to pass through at different times. And that's how your body is going to control the charges within these different neurons, that these channels will open and close, become permeable at certain times so things can pass through them at different times compared to others. So this balance is, again, happening through these different sodium or potassium pumps that are bringing these positively charged ions in and out of your axon. So if we zoom up really close to like the wall of an axon here, we can see something approximating these pumps. So there might be certain pumps that are allowing just potassium to leave the eye or the axon. There might be pumps that allow both of them to pass through, sodium and potassium, or there might be pumps that are just allowing sodium to move. There are different pumps for different purposes, and they all kind of work in concert to change the polarization between the outside and inside of your axons to get this to happen. So the extracellular space on the outside of your cells, outside of your axons, and the intracellular space inside. So this, in the resting potential, inside this darker area, is about negative 70 millivolts compared to this outer area. But to send a message, your axons become just ever so slightly more positively charged. And this signal, this positive, relatively positive signal, is called the action potential. So remember, when they're resting, they're not sending anything, that's the resting potential. An action potential is the signal that is being sent down the axon. And it's not like you can have a slightly different strength here. It's either the neuron sends the message or it doesn't. There's no like weak message or anything like that. It's just that it gets activated or it doesn't. So that's what I mean by all or nothing here. You either have a message or you don't. So if your cell body is sending the message that you're should be charging more positively, the threshold to send that message, remember the resting potential is negative 70 millivolts relative to the outside. The threshold to send a message through as an action potential is just 15 millivolts different. It's still negative, it's negative 55 millivolts. I'm not going to necessarily ask you these exact things, I'm just trying to show you how minute this difference is, these tiny, tiny little changes in polarization and measured in millivolts. So to zoom up on this just one more time with another diagram to show you how this might happen. This change in charge kind of zooms down your axon based on how these pumps are working. So it might be that certain pumps are letting in positively charged sodium ions to change this part of the axon to be slightly more positive. Then the next pumps are going to open up and let that sodium in, and at the same time the pumps where you were previously positively charged are opening to let out potassium, so it becomes more negative. So it's, again, not just like a charge going down a wire, but it's these very slightly more complex electrochemical changes that are moving down the axon by the flow of these different chemicals, these different ions that are flowing in and out of your neurons. So I think after hearing something like that, you're probably thinking, okay, well, that seems fine, but what does that have to do with anything that's actually like happening in our bodies? And I mean, the big answer is that that explains pretty much all of the communication inside of our bodies. Well, we'll get to the endocrine system and how hormones figure into this whole thing too, but neurons explain the majority, we'd say, about the communication between our bodies. The fact that these sensations or these signals also are in this all or nothing state, it also helps explain why sensations don't kind of fade away. Like when you feel pain in your foot, your brain registers that pain with relative accuracy, right? And if our bodies were just using electrical charges, like when you put an electrical charge through a wire in your house, that charge fades over time the further it goes. That's why you need relay stations and transformers to up the charge through wires throughout a neighborhood or throughout a house, because those electromagnetic charges fade. That's not true for the electrochemical charges in our body. They stay exactly as strong because of that chemical process. So this is why we're able to sense things at their kind of proper strength, I guess you might say. But this also, it explains how these signals can run through all these different neurons in kind of this cascading process. Remember when I said that neurons might have 10,000 connections to other neurons? You can see that this process kind of spreads all through these. I'll talk about that more in a minute when we talk about the communication between different neurons. And another maybe more concrete example that might be helpful is there are certain chemicals, certain drugs, or medical tools or whatever that we can use to manipulate this. So certain painkillers like Novocaine or benzocaine or lidocaine, those types of chemicals, the reason they work is because they are, when they're injected, they plug up those sodium gates. So they're not allowing a charge. That's why you get a numbing feeling, because you're no longer sensing, maybe at the dentist if you have Novocaine, you're no longer sensing input from those sensory neurons. And it's because that Novocaine is plugging up those sodium gates and it's not allowing that charge within those neurons to change. So they can't send any information, so you can't feel pain, and you can't send information to move with those motor neurons in that area. So hopefully that gives you an idea of how some of this electrochemical stuff relates to what we're talking about. Okay, so all that stuff is about how neurons are communicating inside one neuron, like the message down from a dendrite and cell body through an axon to the axon terminals. But that's only part of the story, right? We need to also see how the neurons are communicating between one another. So interneuronal communication. This is a really blown up picture of the end of a single axon terminal. So if I'll go back and I'll show you like all these little axon terminals at the end of this little tree-like structure here. Each one of those in the end looks something like this. And it has all these little bubbles of chemicals inside of it, these things called synaptic vesicles that are just little packages basically of neurochemical messengers called neurotransmitters. And when that action potential goes down a synapse, when that action potential goes down an axon to the terminal button of the axon to this space called a synapse between an axon and the next cell's dendrite, it releases these seminal vesicles. Sorry, no, that's a different part of biology. Synaptic vesicles, rather. These little neurotransmitters come out and they go through this space called the synapse between the axon and the next dendrite. And those neurotransmitters are then they're uptaken basically. I can't think of the correct word. They're captured, I guess, by these dendrites. And if they get the right combination of those neurotransmitters to the dendrite, then that dendrite is going to say, like, hey, let's send the message on, basically. So we might think about neurons as this is the presynaptic part. It's the end of one neuron. And this is the synapse right here, the synaptic cleft. And this neuron, the dendrite, in this exchange is the postsynaptic neuron. But basically every neuron has a role that is presynaptic and postsynaptic, right? It just depends what you're looking at. If you're looking at the dendrite of this neuron way back here, then that might be the postsynaptic part of an exchange. So again, once the action potential reaches there at the end, it releases these neurotransmitters. And these are just little chemicals that activate these special receptors in the dendrite, in the postsynaptic neuron. And it's important to note that there are all these different neurotransmitters, and there are all these different receptor sites. And it's a useful metaphor is like a key in a lock. The right kind of neurotransmitter needs to get to the right kind of receptor. If you have a serotonin molecule, a type of neurotransmitter, and it tries to go into a dopamine receptor, that key is not going to fit into that lock. So it's not going to trip that sensor. So it needs to be the right type of neurotransmitter in the right type of receptor. And different neurotransmitters in different locks or receptors are going to have different responses. They're either going to have excitatory responses that raise the likelihood that an action potential is going to be sent down the next neuron, or an inhibitory response that is going to reduce that likelihood or make it further away. So if you have an excitatory response, it's going to cause channels in that postsynaptic membrane down here to open, and positive sodium ions, remember we saw that in the last one, it's going to allow those to flow in to the neuron and increase the charge in this cell. So if you get enough of those excitatory responses across those different dendrites, then you're going to get from that resting potential of -70 relative to the outside to something around -55 millivolts relative to the outside. So you're adding up all these signals from these different dendrites. But it's not just these positive responses from excitatory responses. Some neurotransmitters decrease the charge. So you're adding up all these positive excitatory responses and all these negative inhibitory responses to determine whether a given neuron is going to send its message down to the next neuron. So the sum of these responses is going to determine whether a resting potential goes to an action potential. Again, you need to think about the right key or to the right lock here. So a serotonin molecule, again, a type of neurotransmitter, in some receptors, it's going to be excitatory, and in some receptors, it's going to be inhibitory. It just depends on the combination of these receptors and neurotransmitters. So you can see how complex this gets, that you have all these different neurons sending all these different messages to the next neurons through different neurotransmitters, and if they hit the right receptors, they excite it, and if they hit different ones, they inhibit the response. So there are, literally billions, well, not even billions, there are probably billions of trillions of combinations of neuronal activation that can happen. Okay, just to mention a couple different types of neurotransmitters to give you an idea of what these might be like. Again, I'm not going to ask you super hard questions about these individual ones, but just to give you some examples. One called acetylcholine has lots of different roles, but one of the main ones is that it is going to communicate between your motor neurons and your muscles to actually tell them what to do. So it's going to more often than not have excitatory messages that are going to lead to muscle contraction for the most part. So some neurotransmitters have really specific roles in your motor function and your movement. Some that you probably are more likely to have heard of, dopamine and serotonin. Dopamine is more often associated with inhibitory effects, and this helps you kind of stabilize communication that's going on. Dopamine helps kind of refine the messages that are going on in your brain to make sure there's not just too much noise happening. So if you have the proper amounts of dopamine, you might move smoothly, sleep soundly, that should say, and maintain your posture. But if you don't have enough dopamine, then your movements might be more jerky, and your posture you might not be able to maintain it as well. Serotonin is one that we know has a lot to do with mood stability and how aroused your different physiological components are, and again, regulating your sleep. We know that serotonin has a lot to do with mood because we can use different medications frequently to help stabilize your levels of serotonin, or save serotonin, as it were. We'll talk about that more when we get to treatments for disorders. But for example, selective serotonin reuptake inhibitors, SSRIs, are trying to save the excess serotonin that's not uptaken by, or that's not received by a dendrite, and make sure that it's not kind of sucked back into the axon. So that's one way that people might be able to use medication to reduce the impact of depression and depressive episodes. And finally, a GABA neurotransmitter is one that has to do with relaxation, anxiety, arousal. So GABA, for example, is something that alcohol or benzodiazepine, like anti-anxiety medications, do, or affect rather. So you might have increased GABA if you have had a few drinks, or if you've taken like a Xanax or something. That is what's happening, and that's why those medications have kind of similar effects to alcohol, because they're affecting those same neurotransmitters and receptor sites. Okay, in the next videos, I'm going to start talking about the communication network more broadly, and then we'll get to the nervous system and specific things about the brain.